Hello everyone, I'm Patrick Kunzmann from the Technical University of Darmstadt, and I would like to present you the Python package Biotite for general analysis of sequence data and macromolecular structures. And now you might wonder, why not simply use BioPython for this? And this is actually where the story starts. In 2017, I found BioPython on the internet, and although I really like it, it lacks modern standards for scientific programming in Python, especially consequent usage of NumPy. Therefore, I proposed a modernization of BioPython into BioPython 2.0 on the BioPython mailing list. And the response was quite positive, but since almost a complete rewrite would be needed, there would be issues with backwards compatibility and some possible name ambiguity to which code base the name BioPython refers. Therefore, I continued the project I continued the development in a separate project called Biotite, which has the same license as BioPython for possible later exchange. And then a few months later, I released the first version of Biotite onto PyPI, and a few days ago there was a current release of Biotite, so it's still in active development. Biotite is separated into 4 subpackages that represents a common workflow in computational molecular biology. At first, the database subpackage is used to download data from biological databases like the ISCB or NCBI. The downloaded file are then loaded into the Python object via the sequence or structure subpackage respectively. And based on the internal objects, you can do some, can apply some analysis or editing tools on this. And for methods that are not directly implemented in the Biotite package, there is the application subpackage for seamless accessing external software like NCBI Blast or Cluster Omega. Here's a small demonstration of such a workflow. In this case, we want to align 2 protein sequences of avidine and streptavidine, and for this we download the 2 sequences as FASTA files from the NCBI database. We load the sequences from the FASTA files, align them, and finally this block is for plotting this alignment with the help of matplotlib. One core feature of Biotite is the relatively high performance. Biotite uses Biotite objects internally are based on NumPy arrays, as already said. Therefore, the operations on them are vectorized and C-accelerated. And further acceleration where vectorization is not possible is done via Python code. For example, if you look to the right here are the pairwise atom distances in the structure measured. And in Biotite, this is quite fast because all coordinates are stored a single NumPy array, whereas for example, in BioPython, you have to iterate over each single atom instance. And this is the reason why it's a lot slower in this case. But there's still a lot of work to be done. There's a functionality I want to implement into Biotite, and for this reason, I would like to cooperate with the BioPython people to exchange some code. Yeah. Thank you for your attention. I have a poster, so if you're interested, meet me there or go onto the website. There's an example gallery where you get a small impression what you can do with Biotite. Thank you.
Excellent. We've all kept time. I think we have time for 1 or 2 questions. We have a question online, which I think may have been from an earlier talk. Oh, good, right. Would VCF have applied to the Google talk?
I think that was right.
All right, will you take lessons learned from schema design back to the VCF specification maintainers?
Yeah, so We are also doing some work with that. Somewhat related, we'll be hosting a Birds of a Feather this afternoon, discussing some options about variant schemas moving forward as well. So if you have questions there, definitely come discuss that, but we are feeding that back in.
Thank you. I'll do a general one now. Will the slide decks be shared somewhere? We're encouraging all our authors to submit, for example, to the F1000 channel, or if you want to use Figshare, whatever. And we will be adding links to the BOSC program as and when we get those. Another technical question for Rome. Does it support the Common Workflow Language open standards? Do you support the Common Workflow Language, which is an open standard?
No, we don't at the moment support CWL, sorry. Yes, we do, yes.
And I think one final question. How tight is the dependency between Google's variant transform and BigQuery?
So that is still a fairly tight part to facilitate that. Open to other options there, but have not found any other comparable sources for doing that sort of querying outside of BigQuery. So it does support standard SQL, but a lot of the dependencies of what we do with those queries end up performing a lot better with BigQuery.
Thank you. Thank you to all three of those speakers. We're now going to have another 3 lightning talks. And is Andre ready? So thank you again to those 3 speakers.